Good afternoon, this is Joseph Lin, and welcome to the Autonomic Nervous System cholinergic and adrenergic receptors. In this session, we will cover the following learning objectives, define some key terms, compare and contrast chemical and electrical synapses, explain the general role of calcium in neurotransmitter release, compare and contrast the general functions of parasympathetic and sympathetic divisions of the autonomic nervous system, distinguish between the G sub S, G sub I, G sub Q, G proteins, and how they relate to the ANS receptors. We describe the changes to the effector due to stimulation by the sympathetic division. We identify the neurotransmitters of the sympathetic and parasympathetic division. We will describe the mechanisms of the ANS neurotransmitters secretion and removal. We will describe the two types of cholinergic receptors, list their locations, and describe their actions. We will describe the adrenergic receptors, list their locations, and describe their actions. We will describe the systems innervated by both the parasympathetic and sympathetic division. We will describe the effects of common sympathomimetic and parasympathomimetic drugs on these receptors. All right, folks, let's get started. So here is a list of key terms as you go through the recorded lecture. Synapse, action potential, acetylcholine, ligand, G protein, inositol triphosphate, calmodulin, cyclic AMP, DOPA, snare complex, and exocytosis. The nervous system is the body's primary communication and control system. It integrates and regulates body function through the use of electrical activity. The specialized cells in the nervous system are called neurons, and they have a cell body, a long tail, a terminal, and a ton of dendrites that can receive information. You also see some receptors that can pick up information and monitor changes called the stimuli. The CNS includes the brain and the spinal cord, and the PNS contains the motor neurons and the sensory neurons. The two functional divisions of the motor neurons are divided into the motor neuron that is somatic nervous system, which your body can transmit motor output via voluntary skeletal muscle and allow movement. And then we also have the autonomic nervous system, which is going to be involuntary, without the conscious control of our body, that controls your cardiac cells, your heart, your glands, as well as your smooth muscle. Within the ANS, we have two subdivisions. We have the sympathetic division, also known as the SNS, sympathetic nervous system, and the PSNS, which stands for the parasympathetic nervous system. The sympathetic nervous system is also abbreviated as the FFF, the fight, flight, or fright. When you're fighting for your life, running for your life, or scared for your life. And then the parasympathetic division is going to be your rest and digest / slud system. It's allowing you to stay calm, be relaxed, salivate, lacrimate to tear, urinate, and defecate. Here is a snapshot of the autonomic nervous system. You can see that there are some clear anatomical differences between the parasympathetic division, also known as the craniosacral, because that's where the nerve roots come from, and the sympathetic division, which is the thoracic lumbar division, which is going to be where those nerves predominantly reside. So the parasympathetic division has the origination of neurons in the S2 to S4, but also has four cranial nerves that run the show. Cranial nerve number 3, ocular motor. Cranial nerve number 7, facial. Cranial nerve number 9, glossopharyngeal. And cranial nerve number 10, vagus. Ocular motor is a strictly motor-based nerve that allows you to control the size of your iris and the lens. The parasympathetic will constrict it. The cranial nerve number 7, facial nerve, is a mixed, both motor and sensory. And this one controls some of the neck muscles and highway muscles, but it also innervates the pterygopalatine and submandibular ganglion, which controls the releasing of saliva. The glossopharyngeal nerve, cranial nerve number 9, is also a mixed one, which is also motor and sensory. This one innervates the otic ganglion, which is what controls the parotid salivary gland, again another salivary controller. And then the final one, which is going to be your vagal nerve, which is going to be the 75% of all the output from the parasympathetic division, is run by this nerve. Parasympathetic neurons are generally closer to their effector, whereas in the sympathetic division, they tend to be closer to the spinal cord. So that's an anatomically structural difference between the two. On the right-hand side, the sympathetic division, you can see they're pretty much close to the T1 to L2 region, right next to the spinal cord. And noticeably is the neuron setup. In the parasympathetic division, we have a long pre and a short post. Whereas in the sympathetic, we have a short pre and a long post. Notice that the myelination, which is this fat lipid insulating layer, is in the presynaptic neuron. So this signal is very fast as it jumps. And then for the sympathetic, the pre is also fast, but it's a lot shorter. This circular region is called the synapse. This is where one neuron is able to communicate to another neuron. This region is very important because this is how the language and chemical signals are relayed. Likewise, you can also see that there is a synapse as well for the sympathetic division. So the synapse. What is a synapse? Well, a synapse is defined as this juncture, a specific location where the neuron is either connected or really close to that effector. In the CNS, the second cell will be another neuron, whereas in the PNS, the second cell could be either a muscle or a gland. If one neuron is signaling another neuron, the first cell is going to be called the presynaptic cell, presynaptic neuron, and a second cell will be the postsynaptic cell / postsynaptic neuron. So neuron 1 communicates to neuron 2. There are two kinds of synapses, one called chemical and one called electrical. Chemical synapses are the most common kind found in the body. They're narrow fluid-filled gaps that allow two neurons to be close together, and this region is called the synaptic cleft. It's about 200 to 300 angstroms in length. There is a slight synaptic delay, that is the time it takes for the neurotransmitter, the chemical, to diffuse across the cleft and then bind onto some receptor. And that's how the signal can propagate on. On the other side, we also have an electrical synapse. This is where the cytoplasm of adjacent cells are directly connected via these proteins called gap junctions that allow the ions to flow through, and there really is not much of a synaptic delay because the signals are able to cross right over. And as you can see, the gap between the electrical synapse is about 20 to 40 angstroms, which is 10 times smaller. When a neurotransmitter is released from the presynaptic neuron in a chemical synapse, it can bind to a receptor that is either a ligand ion channel or a metabotropic receptor that requires further signaling inside the cell. These ones can result in a change in membrane potential, a biochemical cascade of signals, as well as upregulate or downregulate certain gene expressions to create more or less proteins. We're going to zoom into the release of a neurotransmitter and the transmissive segment of a neuron. So at the tail end of a neuron, so let's say you have a neuron, you have the cell body, this is the nucleus. These extensions are called dendrites that extend out of the cell body. You have a long tail, and then you have this axon terminal, also known as the butin. And at the end of these terminals, you have this region where there's an opportunity for a chemical synapse to release a neurotransmitter. The neurotransmitter will diffuse across that small little bit of space and then bind to a dedicated receptor protein. So we'll start by talking about how the neurotransmitter is released. The main activity of this transmissive segment, also known as the postsynaptic knob, is the releasing of neurotransmitter in a controlled fashion. As a signal begins to migrate down to the axon terminal, this signal is called the action potential. The action potential represents a moving of a charge between the outer and the inner membrane of a cell neuron. When sodiums rush into the cell, it creates a slightly more positive value inside the cell compared to the outside, and that imbalance of charges is this moving current called the action potential. It follows this all or none gradient to allow the signal to run down. So as this action potential reaches the axon terminal, it's going to trigger these voltage-gated calcium channels to then open up. Calcium, which is higher on the extracellular side of the neuron, begins to rush in and therefore increasing the calcium content inside the cytosol. There's going to be these calcium sensor proteins called the synaptotagmin, which will pick up this change in charge and cause the fusion of the synaptic vessels to begin to form. There's 3 proteins in the snare complex called the synaptobrevin, syntaxin, and SNAP25, which will then pick up this signal, anchor the vesicle that contains a certain physiological dosage of a neurotransmitter, and allow that to dock onto this membrane. Once the membrane is fused, it will then trigger exocytosis, and then the specific physiological dosage of that neurotransmitter can flood across. When there's a greater frequency of action potential, meaning more signals or a stronger signal, that can result in more stimulation and more release of that neurotransmitter. The quantity of neurotransmitter is also directly related to the amount of calcium ions that enter. So the more calcium ions that enter, that could also create a greater amount of release. This is a flow chart of what happens from first step to the last step. When the action potential reaches the end of the axon, the voltage-gated calcium channels open up. Calcium stimulates the fusing of the synaptic vesicles to the plasma membrane, and exocytosis occurs that releases out the neurotransmitter. Keep in mind that when calcium enters a cell, it binds to this calcium sensor protein called synaptotagmin, which will anchor the vesicle to the membrane. This is not completable without the help of the snare complex proteins. The snare complex proteins included with the synaptotagmin complex will then fuse onto the presynaptic membrane to generate this release site where pores will allow the exocytosis to occur. Roughly 2,000 to 10,000 molecules of a neurotransmitter could be present inside every single vesicle. For example, acetylcholine contains this range of signals.